All right, so next up we're going to be talking about proteins. Proteins are polymers of amino acids that are linked together by peptide bonds. And so the polymer form is going to be known as a protein or a polypeptide. The monomer form is going to be an amino acid. A peptide bond is just a covalent bond between amino acids. And as much as 50% of the dry weight of most cells is going to consist of proteins. And there are several thousand proteins that have been identified. 2 or more amino acids joined together are called peptides. Long chains of amino acids joined together are going to be called polypeptides. Now the interesting thing is that once we have all of our amino acids strung together, what we have is what's known as a polypeptide. It doesn't actually become a protein until that polypeptide is folded up into its correct three-dimensional shape. And we're going to see a lot about the significance of that three-dimensional shape as we move on in our discussion about proteins. So just note that I'm not going to ask you to tell me the difference between a polypeptide and a protein, but you're going to see that terminology a lot, so I just want to help clarify. The polypeptide refers to that sequence of amino acids. It's not actually considered a protein until it's folded up into its correct 3D conformation. But we're going to start off our discussion of proteins by talking about their functions. And proteins have a lot of functions, much more beyond just structural or beyond just muscle tone. And so if you ever see that question about what would happen if you had a diet that was not rich in proteins, or if you suddenly stopped consuming large quantities of proteins, what would have happened to the body overall? A lot is going to happen. A lot can go very wrong if we do not have the correct amino acids. A lot more than just loss of muscle tone or muscle definition. So some of the functions of proteins are going to be 1, metabolism. Almost all, and if you remember if you remember, what does metabolism mean? Metabolism is going to be the sum total of all the chemical reactions in the body. So everything from the digestion, breaking down of glucose into cellular fuel ATP, or the building up of muscle cells, the building of or the building of your skeleton, or the secretion of hormones, the picking up of hormones, the signaling that happens. All of these are regulated by metabolism. So most enzymes are going to be proteins. They will act as catalysts to help accelerate those chemical reactions within the cell. So that's the role that proteins play in metabolism, is by acting as enzyme catalysts. Proteins are also heavily involved in support. Some proteins will have a structural function. For example, keratin, collagen make up essential components of our skin and our fingernails or our hair. So they'll have sort of a structural function. Proteins are also involved in transport. Membrane channels and carrier proteins are going to regulate what substances enter and exit the cell. So when we're talking about transport, we're talking about cellular transport. How do we get molecules into and out of the cell? Hemoglobin is the protein that transports oxygen to tissues and cells. It's a major component of our blood. And again, that's all a protein. Proteins are also involved in defense. Antibodies are proteins of our immune system that bind to antigens and prevent them from destroying cells. So they play a very important part in our immune system. They're also used in regulation. Hormones are regulatory proteins that influence the metabolism of cells. There are many hormones that are steroids or lipid hormones, steroid hormones, but there are also a great deal of our hormones are actually going to be proteins, and they're going to play very important roles in the metabolism of cells. And then finally, motion, microtubules. And again, proteins do play an important role in our skeletal muscles. They do play an important role in our skeletal system and allow the body to move as a whole, but they also play a very important role in motion on a cellular level. Microtubules are going to be part of the cytoskeleton, and they're going to help move cell components to different locations. Actin and myosin contractile proteins are going to be responsible for muscle contraction as a whole. So there are a lot of different roles that are being played by these proteins, and I want you to understand or appreciate just how varied those roles are. We're going to talk a little bit about proteins structurally. Amino acids are going to be the building blocks that are necessary to construct proteins, and all proteins are made up of only 20 different amino acids. And with just 20 amino acids, we get all of the different diversity, all of the structural diversity between every living thing comes down to ultimately the sequence of those 20 different common amino acids. Amino acids are going to vary by what is known as the R group or the variable group, which ranges in complexity. All proteins have same basic structural features. They all are going to have an amino group over here. They're all going to have a carboxyl group, an acidic group over here on this side. And bound right in the center, attached to a central carbon, is going to be the variable group, the R group. So this is going to be the part that changes depending on which amino acid that you're looking at. Just as we've mentioned before, we're going to synthesize and break down our amino acids the same way that we synthesize and degrade any of our other large macromolecules. We're going to use a dehydration synthesis. We're going to kick off an OH group from the carboxyl group on one side of the amino acid, and we're going to remove a hydrogen atom from the amino group on the other side of another amino acid. Sorry, words are hard. But together, we're going to remove one hydrogen, one hydroxyl group. We're going to produce a peptide bond, which is just a covalent bond between two amino acids, and kick off a water group. This is what's known as a dehydration synthesis. So the next thing that I want to talk about proteins, we kind of already mentioned that proteins aren't proteins until they are in their correct three-dimensional conformation. And so when we're talking about proteins, it's important to understand the distinct levels of structure that are ultimately going to contribute to their function in the body. And fortunately, these structures, or these four distinct levels, were named by scientists. And as we are going to find out through the course of this study, is that scientists aren't necessarily a very creative bunch. They tend to name things very straightforwardly when they have the chance. So the four levels of structure of proteins are going to be the primary level, secondary level, tertiary meaning 3, and the quaternary level, meaning the 4th level. So we're going to start off by looking at the first level of protein structure. The first level of protein structure is going to be the primary level. The primary level is going to be a linear sequence of amino acids. So this is going to be one amino acid here, another amino acid here. This could be arginine, leucine, isoleucine, proline, valine, arginine. But it's just going to be the sequence of amino acids, those 20 different amino acids that we've mentioned. And now there are hundreds and thousands of different types of polypeptides that can be built from just 20 amino acids. It's a lot like the way sentences can be made from any number of words, or the way words can be made from just 26 letters. So think of proteins as words. We have 26 letters in the English language. There are 20 amino acids in life. And with those 20 amino acids, you can put them together in any combination of any length to come up with any word or any type of protein. So that's going to be our linear sequence of amino acids. This is what's known as the polypeptide chain, as we've already mentioned. That sequence of amino acids is going to be our peptide chain. When we change the sequence of amino acids, we're going to produce different proteins, or each time we change that sequence of amino acids, we're going to get a different polypeptide chain. Now, this sequence of amino acids is going to be coded for in your DNA sequence. So I don't know if you realize that, but when we talk about DNA being used to store information, all DNA encodes for is amino acid sequences. The information that's found in your DNA is used to create proteins, and those proteins can build up, tear down, or make any number of features that ultimately result in you as a person. So that is what your DNA is coding for. Your DNA is going to be coding for that sequence of amino acids. And one way to help you kind of remember that primary structure is that this is the primary level. This is the one-dimensional level. And a single-dimensionary object is going to be just a straight line. So this you can think of as 1D, linear. Whoops. For some reason, the slide is not progressing. So, all right, our second level of protein structure is going to be the secondary level of structure. This is where we're going to see that line become a shape. And so we're going to see that we have we're going to see either protein coils or folds. It's going to be what's known as either an alpha helix, and that'll be this structure right here. So here is our long chain of amino acids. It's going to be wound around itself in what's known as an alpha helix, or it's going to be folded back and forth, back and forth, and back and forth. And it's what's known as a beta pleated sheet. You can kind of think of this as corrugated cardboard with that folding back and forth and back and forth. This structure right here, the secondary level of structure, is going to be held together by these red dotted lines. Where have we seen these red dotted lines before? Does anybody know what these red dotted lines indicate? If you said hydrogen bonds, you are correct. This is another location where we're actually going to see hydrogen bonds. Yes, hydrogen bonds are responsible for holding water molecules together. They are also responsible for the secondary level of protein structure. So each time you see these red dotted lines, you can think of hydrogen bonds that are formed between the partial positive and the partial negative charges of the different amino acids inside of this sequence. You can also think of this as the two-dimensional level. We have gone from a linear sequence, we've gone from a straight line, now we have a shape. We have these coils or this pleated sheet. So it's our two-dimensional structure, our secondary level of protein structure. Our next level is going to be the tertiary structure. This is going to be the overall three-dimensional shape of that polypeptide. This is where we go from being just a single two-dimensional structure to a three-dimensional structure. So this is ultimately going to be stabilized by the interaction of the hydrophobic amino acids within the polypeptide to help fold and maintain their structure. So if we have, you can see right here, we have an alpha helix. Here we have a beta pleated sheet. Here we have an alpha helix. But what we'll find in this long sequence of amino acids is that some amino acids are hydrophilic. Some amino acids are hydrophobic. Some are charged. Some are neutral. All of these amino acids have different characteristics. Those amino acids that are hydrophobic are going to be found where? We're going to see that our amino acids that are hydrophobic are going to orient themselves towards the inside. They're going to be sort of right here. We might have a hydrophobic amino acid, maybe one right here, right in here, right in here. Our hydrophilic amino acids are going to want to be on the outside. They're going to want to be near the water. So we're going to find hydrophilic amino acids maybe over here or right over here. Right over here. That sequence of hydrophobic or hydrophilic amino acids is going to determine that three-dimensional structure. They're going to help pull that straight line into a three-dimensional shape. So the nonpolar amino acids tend to fold together to keep away from the water surrounding the molecule. So consequently, the hydrophilic amino acids tend to orient themselves facing outward. And these chemical interactions, as well as the hydrogen bonds, the ionic bonds, covalent bonds between the R groups, are going to help contribute to that overall tertiary structure of the protein. So that's going to be our three-dimensional structure. The last thing that we're going to say, or the last level of structure, is going to be the quaternary structure. Some, not all proteins have a quaternary structure. This is where we're going to see, the quaternary structure is when a protein consists of two or more polypeptides. A great example of this is going to be hemoglobin. Hemoglobin is a globular protein. It consists of 4 different polypeptide chains. Each one has its own primary, secondary, and tertiary structure. So in this case, we actually have 1, 2, 3, 4 different polypeptide chains, and they all are going to come together to form a larger quaternary structure. And again, not all proteins have a quaternary structure. Some do, some don't, but all are going to have a primary, secondary, and tertiary. For a great summary of this, check out figure 2.22. It's going to walk you through the primary, secondary, tertiary, and quaternary structure. I would advise, just as a study aid, that you use this table. Cover up, put a little sticky note over all of these words here, and see if you can just walk through and explain, or draw through and explain all of the different levels of protein structure. If you can do that, you've got an excellent grasp of what you need to know for protein structure.